All right. So let's now talk about degrees of dominance. Okay. We've been talking about what's called before when we discussed Mendelian genetics. Remember, watch the first video first. Mendelian genetics and what Mendel did was normally dealing only with complete dominance, but Mendelian principles can be applied to incomplete and codominance, which we're going to talk about. Complete dominance is when the heterozygote and the dominant homozygote are identical. It doesn't matter if you have a big B or a big B little b, everyone still has brown eyes. You can't distinguish them. There's no difference. In incomplete dominance, the phenotype of the F1 hybrids is somewhere in between, okay? Instead of making purple and white, you make red, pink, and white, which means that the heterozygote actually looks different than the homozygous dominant. It means that the dominance doesn't quite come out. The reason why skin color is the way it is, where you actually are often a combo of your mom and dad's genes, it's because that combo is each of the 20 genes or so that control your skin color contribute a tiny bit of color. So depending on what combo of genes you have, you're going to get the tiny bits of color from everything. It does look like it's blended, okay? That's because of incomplete dominance. In codominance, the two dominant alleles affect the phenotype in separate, distinguishable ways. We're going to talk about both of those. When dominance is incomplete, all genotypes have their own unique phenotype. Snapdragons, for example, their phenotypes are red, pink, and white. You have two alleles, not three. One allele for red, one for white. Heterozygotes are pink. So here you have a homozygous recessive. Well, not even recessive because it's so homozygous white, homozygous red, heterozygous. One of each. Looks like a combo of the two, and that's because of incomplete dominance. So here you have, again, note these are not labeled with dominant and recessives anymore because neither one is truly dominant or recessive. You have the red 1, which two combos of the red 1 will create red, and R2, two combos of that will create white. Okay? The parental generation or true breeding, okay? Homozygous, homozygous. Creates a heterozygote. With one copy of the red and one copy of the white, you create pink. So when you cross two pinks, what is the ratio you're going to get? So if you have a minute, stop this for a moment, make yourself a Punnett square. So you're going to put, you can do big R, little r. Imagine the big R is red, even though it's not fully dominant. Big R, little r, big R, little r. They're heterozygotes. What ratio will you see? So see if you can do that, and what ratio do you expect? The answer for that, a 1 to 2 to 1 ratio. Remember, in any Punnett square, you're going to get 1/4 homozygous dominant, 50%, it's going to be 50% recessive, 50% heterozygote, and 25% recessive. But remember that because it's incompletely dominant, these two are the in-betweens instead of looking directly like the dominant, because it's not fully dominant. This is called incomplete dominance. Okay, so this shows you the actual setup here. Note that you have the color red and color white organized in this way instead of having the color, because it's just a color gene now. It's no longer a actual dominant, not big C, little c. Okay. All right. So that's your incomplete one. Let's talk about codominance in multiple alleles. This gets tricky. but not too tricky. Codominance means that the two alleles of a single gene produce two gene products. And instead of one being a blending, you have a joint expression of both alleles. Both alleles come out. Both alleles are expressed. There's no dominance, there's no recessiveness, just both of them express a different protein. For example, in sickle cell, in the heterozygotes, and you probably know someone who is a carrier, has the sickle cell trait, but not the sickle cell disease. The sickle allele creates sickled cells. The regular one, which for anyone who is not a carrier, you only have these normal blood cells, okay? But some of them will create normal blood cells. And so people with the trait have sickled and normal, which means both of them are expressed. Neither one dominates. If you have one copy of the trait, you don't have the disease, you just have partly sickled cells. So that would be considered a codominance. For instance, if you have white and black come together, note they're labeled with two different letters. The reason is this is not a dominance effect at all. These are two different genes producing, they're the same gene, sorry. Two different proteins are created by each one. Note that the heterozygote does not look gray. It is instead black and white. That's the difference between incomplete dominance and codominance, okay? All right. So you see here, if you have two sickle cell alleles, you create only sickle cell hemoglobin proteins. You have sickled cells, and you have sickle cell disease. Meanwhile, if you have one sickled and one normal, you create both. And you have a sickled, half your cells are sickled and half your cells are normal, but you don't have as much of an issue here, okay? So you have some symptoms of maybe your oxygen is low. So someone would say the sickle cell trait would be more susceptible to COVID due to their inability to deal with lower oxygen levels. Okay? All right, next concept, multiple alleles, codominance and multiple alleles. This is one of the key things to talk about is the ABO blood type, because everyone has some, you probably know your blood type, okay? My blood type is O. O is recessive to A and B. A and B are codominant. 4 phenotypes of the ABO blood group in humans are determined by 3 alleles for the enzyme. You either have an A carbohydrate, a B carbohydrate, or no carbohydrate, which is my case. Okay? The enzyme encoded by the A allele as the A carbohydrate. The enzyme encoded by the B allele as the B carbohydrate. The enzyme encoded by the I allele adds nothing. You can have both A and B. That's the AB blood type. The I allele, it's recessive to both. And so if you have 2 I alleles, which I do, you get O blood. We're not going to worry about positive, negative right now, okay? Put it out of your head. All right, so here's how it looks. Again, there's a carbohydrate here, carbohydrate here. If your genotype is AA or A O, okay, so that's the O, but A nothing, your red blood cells look the same, okay? This one doesn't add more carbohydrates on just because it has 2 copies. Same number of carbohydrates. Your phenotype is A. B can either be this one or this one. Only as the B carbohydrate, your phenotype is B. AB, everything. AB blood type. So you have both A and B, distinct, different expression. O, I have no carbohydrates. None. Okay. Here is a cross now. To do a cross for this, what you have to do is actually figure out what everyone has to give. And then you'll be able to figure out what the possible blood types are. Susie's blood type is O. Her husband Sal is type AB. Alright, so let's start with O. O. She can only give little I or little I. Okay, the O type. Sal can give A or B. So put an A with an O, or put a B with an O. The only options are A, the genotype would always be this. B, the genotype would always be this. You can't be type O. Susie only has an I to give, and the O genotype is little I, but Sal has no O's to give. He can only give A or B, therefore there is no way to have a O child, because Sal doesn't have any O's to give. Okay. It cannot be AB. Actually, I can never have an AB child. I can't give A or B. But in an AB child, A from mother, B from father, or opposite, okay? A from father, B from mother, okay? You can't express both in the offspring. So, because Susie's always going to give that O. And so that's an interesting concept, right? All right, let's now move to how paternity can be told by this. Howard Hughes has type AB. This is a true story. A woman claiming to have had his child. She is type A. The child is type O. Is this his child? Remember that O has to have a O from each parent. AB has all their slots filled. There is no O that can come from Howard Hughes. He can never create an O child. His child could be type A. His child could be B. His child could be AB. A from mom, B from dad. Since she has a child who's O, she also has an O to give. The only thing it cannot be is O, actually, because only one parent has an O to give. Again, figure out who has what to give. Howard has AB, so one allele is A and one allele is B. There's only 2 allele slots. There are 3 total alleles, but no one has all 3. One allele is A, one allele is B. Woman is a O, or that I. She gave birth to and must be the mother of an O child. She has a hidden O allele. She can give an A or an O, but she cannot give a B. Child is OO, the little I. Must get an O from the mom and dad. The supposed dad has no O to give. Howard is not the father. Okay. So in order to work this one out, let's do an actual one that I know. Let's work through me and my husband, okay? So let's just go copy this one and delete, okay? Let's do this. I'm O. I've always been O. I know I have to be O. A, because I've given blood several times. B. My mom is O. My dad is O. They have nothing but O to give me. So I am I. Or sometimes it's expressed as OO. Okay. My husband. He is A. Before we had our child, we didn't know what his full genotype was. Let's look at his parents. Do we know what his blood type is? The answer is we don't. Because we just thought he'd be A. AA. Came from both sides. Maybe he's big A, right? My daughter. So what is my husband's genotype if he created a no child? So this is where you can figure out, what can my husband give? We know, I'm telling you, my daughter is a carbon copy of my husband. There's almost no way. In the sense also that obviously we know that this is our child. So here we have a child that is O, and my husband is A. But if this is his child, then it must be that he husband must have an I to give. Therefore, let's do it in the proper way, just so I don't confuse all of you. Did this before. This needs to be up, but I don't know how to make it do that. Not gonna do that here. That's fine. Let's just copy that. My husband is IA. That has to be his genotype, because he cannot have an O child unless he has an O to give. Since he's not AB, we knew that it was likely that one of his parents, either his dad or his mom, one of them had to give A to him. Either his dad or his mom gave him a recessive O allele. There are problems in here that have to do with that, okay? So let's go look back and look at, I already worked at a few of these in the previous one. All right. This gives you similar concepts, okay? One mother has type O blood, her partner is type AB. The other mother has type B blood, the partner is type O. So you need to understand, if the babies were mixed up in the nursery, would it be possible to assign them to the correct parents based on blood type alone? Okay, I'll work through this one, but we talked about these in here, okay? All right. So let's talk about this one, okay? 2, baby A, okay? Mother has II. Father, AB. Child, A or B. Okay. Baby B. Mother, B, question mark. Father, O. Okay. So, it's the AI or BI, okay? Baby. Now, we can do a Punnett square here, but we can also just figure it out, okay? Let's say the mother is B. The baby would only be able to be B or O. Because remember, this could be a hidden B, a hidden O, or it could be a B. The deal here, if one child was A or O, they could be assigned to the correct parents. You know that an A child can only belong to the first set of parents. You know that a B child could only belong, an O child could only belong to the second set of parents. But if both were B, you could not. Okay? Because both of these have an option to have a B child. You'd have to figure something else out. And that is a negligent hospital, just saying that. It depends on the blood type here, okay? Then obviously, we've already answered some of these questions, but I want you guys to think through them, okay? And please explain why or why not. We talked about A, B, and O, Howard Hughes. Talked about O and AB. I'm O. Can I have an AB child? And my husband's parents were both A. Could they have had an O baby? Remember that you don't always know the genotype if you only know A. You know the genotype for AB, you know the genotype for O. A and B have two different genotypes. All right. We have this one. It's very similar to the Snapdragon problem. I want you guys to work it out via Punnett squares. Okay? That's all we have for this week. So I'm going to end this, but remember to come to one of the sections where I discuss this. Working through and practicing these is one of the few ways to actually get it down. I'm not going to include any of these problems on the quiz number 2. But I want you to be able to figure it out, okay? So just work through them, practice, and do the ones that I gave you, especially the Mendelian monohybrid ones. Figure that out, okay? All right. I'm going to stop recording now. But again, please come, and we're going to do lots of practice. I have a whiteboard in here, so we can work on the whiteboard.